we had left our biology heroes with the first three things that we would talk about as causes of increased antibiotic resistance. Use of antibiotics inappropriately, people who don't finish the course of antibiotics, and antibiotics in livestock. This fourth one is probably the easiest one for you personally to do something about. I'll read it, then I'll shut up and let you copy. Causes of antibiotic resistance: antimicrobial, antibacterial soaps and other products. Antibacterial soaps contain low doses of an antibiotic, usually triclosan. After antibacterial agents are applied to a surface, your hands, the floor, the counter, they stay there and continue to kill bacteria. Which bacteria do they kill? The weak ones. Who does that leave to reproduce? The resistant ones. Antimicrobial chemicals in these products act in a similar way to some antibiotics used in humans. Okay, so the biggest issue here is that antimicrobial products that contain an antibiotic give bacteria lots of practice with that antibiotic. If you take the same test 3 times, should you score better the 3rd time? Yeah. So 3 generations of bacteria exposed to the same antibiotic, the antibiotic becomes less effective with every new generation. In general, that's typically what happens. The problem with antimicrobial substances in the house and in school and every place else is they stay on that surface. They keep exposing bacteria to those antibiotics. And eventually you just kill off all the weak ones. Who here has ever used Neosporin on a cut? Triple antibiotic ointment, little tube, somebody keeps it in the medicine cabinet, you get a cut, you put it on it. It's a miracle. It keeps things from getting infected. Fantastic. What would happen if you used it as a moisturizer every day? I'm going to get this giant-sized vat of Neosporin. I'm going to cover my entire body with it every day. What's going to happen? What's going to happen? Sawyer? It'll stop working on cuts when I actually do need the protection of that antibiotic, because every single bacteria on my body surface, and there are a lot of them, everything that's actually able to be killed by Neosporin is going to die. And the only things left are going to be those things that can tolerate Neosporin. And then I can up the dose, and then who am I going to leave? The ones who can tolerate a double dose. So this is an issue. A friend of mine, a very good friend of mine, is a lab technician at the Cleveland Clinic in downtown Cleveland, Ohio. Do you know what a lab tech does? Okay. Has anybody here ever had blood work done or been in the hospital or had any kind of testing done? So, me too. Somebody draws a sample of blood, somebody has a urinary tract infection, they pee in a cup. That sample goes to the lab. And my friend Katie has been doing this for 25, 30 years. She counts cells. She gets your blood sample. She runs it through a machine. It pops up on a screen. It analyzes the number of red cells, white cells. Somebody else in the lab does chemical testing. They can look for the presence of various chemicals in your blood. Somebody else usually does the cytology testing, where they're looking at a smear from that urine sample and they're looking for particular bacteria in your urine. That's what she does all day, every day. And it's an occupation with a pretty high risk of exposure to dangerous bacteria. Number one, she's working in a hospital. Most of the people who've gone there are sick. Ew. I don't want to be around a bunch of sick people. Number two, if their stuff, their blood, their feces, their urine, their whatever, because she deals with all of that, is being sent to the lab, it's because there's something wrong with them. There's probably something abnormal and unhealthy in their blood, feces, or urine. So they have, she's talked about it for years. she has scrubs that she wears at work. She wears white scrubs. She says they can wear like 3 different colors. She said, I wear white because stains show up and I sure as all get out want to know if something got on me. I want to know immediately. Seems reasonable. She gets to work in her street clothes. She changes into her scrubs. Before she goes home, those scrubs go in a bag. At home, they get washed in bleach and hot water. But I think she does have to wash her own scrubs. She does not wear those home. They're bagged up. They go straight into the laundry. She doesn't want to carry stuff home on her clothes. She is handling these samples. Now, she's gloved every day, but she is very, very careful. They have an extensive protocol for cleaning their workstations. And we've talked about, how do you kill, because what would happen if they were using an antibiotic on that work surface? You could develop bacteria in the lab that were resistant. So what do you want? Here's what we need to talk about, is that there's more than one way to skin a cat, kill a bacteria. So, whoops, where'd my little friend go? There's my little friend. There's my little friend. There's your little friend. There's our little friend. Maybe not such a friend. We don't know what this one does. So if we assume that there's some variation in this population of bacteria, and I'm just going to do a representative sample, and these guys are all pretty similar, but I'm going to take one of them and make it a little bit different. Oops. Let me give you a slightly thinner protein coat. And I'm going to give you a protein coat that has extra folds in it. So same protein, but this one has sort of like specialized folds in it. So it's a little bit thicker because of that. It's got sort of these jaggy things. There. Okay, so we've got shaggy, we've got, a couple others. Copy, paste. And this is where we also get into that idea that resistance isn't always total. Resistance is not always 100%. And we saw that with our, we saw that with the little exercise we did with the Skittles. Even those resistant bacteria, eventually, given enough antibiotic, often enough, you could start to kill them. But it's harder. So here we have the same species of bacteria, 3 slight differences in their protein coats. If we apply an antibiotic that very specifically targets a chemical in their protein coat, and it targets it because of its thickness, we may kill all of these guys, and all of these guys, and maybe even all of these guys, but we're not touching these. But there are chemicals that are hard to build resistance to. And they're things that typically target all cells. So it's not targeting the protein coat in this cell. It's not targeting some chemical process happening in the cell. It will kill any cell. It will kill anything. It is what we call broad spectrum. Okay? So something that is broad spectrum will kill everything. And we talked about alcohol being fairly broad spectrum. The hand sanitizer said something like kills 98% of, or 99.9% of all germs. One of the things that they wipe their workstations down with at the Cleveland Clinic, because my friend Katie has been looking to alternatives to Clorox, and she looked at rubbing alcohol. And it is effective against 99% of all germs. There is one that it is not effective against, apparently, which is C. diff. It's a gut bacteria. It's often a problem for people who are in chemo or who've had heavy rounds of antibiotics. It lives in your gut. It's fine as long as it's kept in check by everybody else. If you kill off a lot of the other gut bacteria and the C. diff takes over your gut, then you can die. And it's a problem. Alcohol doesn't work on C. diff. Works on everything else, pretty broad spectrum, but it doesn't work on C. diff. A chemical antibiotic that targets specific bacteria is easier for bacteria to evolve resistance to. The equivalent of dropping a nuclear bomb on the bacteria, which is something like Clorox or alcohol that kills almost all species, almost all cells, is harder to develop resistance to because nobody survives to reproduce. No one. And, you know, for things that are antimicrobial for like wiping surfaces, we would prefer to have that. Okay, we're going to look briefly again at that video from yesterday from Harvard, because I think seeing it twice after we talk about the effect of having an antibiotic on a surface that bacteria are constantly exposed to is probably useful.
So we learn about different plants in the fields.